So I sent out the message about exam regrades. I will be doing them this weekend. If you haven't picked them up, that's fine. I will take and regrade all the ones that either people have turned back into me or the ones that people haven't picked up. And I'll put them back in the BB office for pickup on Monday. So should be set there. Any questions that you have or whatever, as always, let me know. Moving through translation, there's a few things I want to say, one of which is on the screen that I meant to mention last time, and I didn't get a chance to do so, so I'll do that here. And that is the actual organization of protein coding regions in bacterial cells. So bacteria are somewhat different, as I've talked about before, much smaller genome than eukaryotic cells. And I've mentioned before that genome is very efficiently organized, very little spaces between genes and things like that. It's even excessively organized in that sense, in that bacteria have something that eukaryotic cells don't have, and it's what we call polycistronic messages. So polycistronic messages means that a given messenger RNA can have more than one coding sequence on it. And that's very commonly done with bacterial genes. So for example, the tryptophan operon has 7 or 10 individual proteins that are encoded on a single messenger RNA. And in the case of bacteria, that organization of genes in a polycistronic fashion is functionally related usually. That is, when you see multiple genes organized together on a messenger RNA, they usually have related functions. So for example, as I say, bacteria use tryptophan as I use a polycistronic message to code for proteins that make tryptophan. Okay? And when that message is translated into protein, all of those proteins that come from that coding all are involved in helping the cell to make tryptophan. There are many other examples, tryptophan being just one of them. The organization of multiple genes in a prokaryotic cell in this fashion creates what we call an operon. Okay? An operon. So an operon is a prokaryotic structure, and literally it means organization of a variety of genes under the control of a single promoter. The single promoter is responsible for making sure that that long messenger RNA that encodes all those proteins is activated. Okay, well, we'll see examples of that when I talk next week about gene expression and how the organization of those multiple genes in a prokaryotic cell really helps to simplify things for the prokaryotic cell. Eukaryotes do not do that. Eukaryotic cells have one gene, one messenger RNA. They don't put multiple protein coding regions onto messenger RNAs, only prokaryotic cells. Okay, I talked about the genetic code, and I also finished at about this point talking about the amino-acyl tRNA synthetases. I said there were about 20 of the amino-acyl tRNA synthetases, one for each gene, for each protein, each amino acid that is used to make a protein. And I talked about how that attachment of the amino acid was called charging, okay? Or activate. Actually, there's two phrases that are used. One is charging of a tRNA, and the other is activation of an amino acid. They both refer to the same thing. In both cases, we're talking about attachment of an amino acid to either the 3' or the 2' end of a transfer RNA. Okay. Now, if we look at the process of translation, okay, we sort of see it schematically here. You can see the amino acids coded for by the genetic code that are there. There is the messenger RNA that was copied from the genome, and there are the complementary anticodons that are found in the transfer RNAs of the transfer RNAs. It's important to remember that all base pairing that's done in cells is done in an anti-parallel fashion. So therefore, when we read a codon in the 5' to 3' direction, we have to think that the anticodon is read backwards to that, okay, because they're anti-parallel. So if I were to look at this 5' to 3' direction for this messenger RNA below, the anticodon above, if I started with this A and went leftwards, that would be 5' to 3', A, G, C, going from right to left. So it's important when you designate any structure that you indicate the polarity. What's the 5' end of what you're talking about, and what's the 3' end? Now, I tell you that because sometimes you will see what I talked about last time was that third position of a codon is rather variable in most cases. I showed you an example of leucine, for example, that the third position could have any of the 4 bases present. Okay? That design that we see in codons refers to what we call the wobble position. That third position is called the wobble position or the wobble base. The wobble base is at the 3' end of the codon, which would mean it's at the 5' end of the anticodon going in the other direction. Well, what's happening in the process of translation is schematically what you can see on the screen. The ribosome binds to the messenger RNA, and it slides along the messenger RNA. Translation as a process in the ribosome solely uses GTP for energy. GTP is the energy source of protein synthesis. Now, that very first step, that attachment of the amino acid to the transfer RNA, that's the only part of the process that requires ATP. Everything else that happens in the ribosome, or everything that happens in the ribosome, requires GTP. So GTP energy is a very essential thing for the process of translation. Another thing to remember about translation is that translation occurs exclusively in the 5' to 3' direction. So you've seen DNA replication, you've seen transcription, and now you've seen translation occurring 5' to 3'. One of the things I did not mention last time, but I will mention here, is I did say, for example, that translation and transcription in a bacterial cell are happening frequently in the same place at the same time. Because there's no nucleus in a bacterial cell, the ribosomes can attach to the messenger RNA and start translating that messenger RNA as soon as it emerges from the RNA polymerase. That places a limit on how fast translation can occur, because translation can't go faster than the synthesis of RNA. And so no surprise when we look in bacterial cells at the rate of translation, we see it's roughly at the rate of transcription. Transcription, you may remember I said, worked much slower than DNA replication. DNA replication was going at 1,000 nucleotides a second. Transcription is going only at about 50 nucleotides a second. And translation is going at about 15 to 20 amino acids per second. Well, if you think there's 3 bases per amino acid, it's going at roughly 45 to 60 nucleotides per second, which is roughly what transcription is going at as well. Now, it's pretty remarkable to think about this process I'm going to describe to you today is going on at that rate. Okay? I will agree with you, it's perhaps not as impressive as 1,000 nucleotides a second for DNA replication. But you're going to see that translation is a fairly intricate process as well, and that it's able to go at this incredible rate that it actually goes at says something, again, about how the world that's nanoscopic is very different than the world that's macroscopic that we live in out here. It works. If it didn't work at the rate it does, then life would be very different than it is. Okay. This simple schematic shows basically what or how things are organized in the process of translation. The ribosome, of course, is the complex of RNA and protein that is where the newly synthesized protein is actually synthesized. And the ribosome contains 2 main components, something we call a large subunit and something we call a small subunit. The large subunit in prokaryotic cells has a size of about 50 S. And when you see that S designation, S stands for Svedberg units, which you don't need to know. But S units are a measure of size. They actually come from the way we studied these originally, which was in centrifugal fields. The larger the S value, the larger the molecule or complex that we're talking about. So the large subunit has a size of 50S. It contains ribosomal RNAs, and it contains proteins, as I will show you. The small subunit is, and by the way, the 50S size is in prokaryotic cells. The large subunit in eukaryotic cells is a bit larger. It's about 60S in size. In prokaryotic cells, the small subunit has a size of about 30S. It also contains proteins, and it also contains ribosomal RNA. And 30S is smaller than 50S. The S units turn out not to be additive, so if you put together a small subunit of 30S and a large subunit of 50S, you get an overall structure of 70S. As I would say, don't do the math, right? Okay. So Svedberg units are not additive, but they're relative to each other. In the eukaryotic cells, the small subunit is bigger also. It has a size of about 40S, and the 40S plus the 60S give an overall size of about 80S for the ribosome for eukaryotic cells. Now, as you can see in this simple schematic, this structure has sort of, as I say, two components to it. I sort of like to think of it as like a snowman, okay? An upside-down snowman. So the snowman, when you make a snowman, it's got a great big snowball at the bottom and a less big snowball above it because it's hard to lift that snowball up, so you put the smaller one on top. And so this is an upside-down snowman, the large guy on top and the small guy on the bottom. For our purposes here, it's important to see those three structures that you see in the middle. These three structures have things labeled various sites, the A site, the P site, and the E site. Okay? These sites are places where transfer RNAs that are linked to amino acids bind. Okay? So, with the exception of the very first transfer RNA, all of the incoming transfer RNAs come in at the A site. The very first one starts in the P site, and we'll see that happening in just a bit. What's happening during the process of translation, as I noted, is the ribosome is sliding down the messenger RNA, and in our case, the ribosome would be sliding from left to right. So that incoming transfer RNAs would come into the A site. We'll see that they will slide to the P site, and then they will slide out the E site. E stands for exit. Okay? And that's happening as the ribosome itself is moving rightwards in this figure. So things are coming in here, going out there. The ribosome is sliding just like this. Okay, so that's the overall scheme that we'll have for translation and the organization of materials in the ribosome. Okay. I love this figure of this transfer RNA. I used it in our textbook because I think it's one of the coolest images. I showed you before a schematic for what transfer RNAs look like in 2 dimensions. And when you look at this structure in 2 dimensions, it's very simple, okay? But in 3 dimensions, it doesn't occur that way. Well, what's the difference between the two? Well, in 2 dimensions, we're able to see all of the base pairing that's happening. And when we first studied transfer RNAs, we knew they had to have some kind of a structure like this one in 2 dimensions because of all the possibilities of base pairing that are there. But when the people first started studying this, they were a little puzzled. Okay? They were puzzled because they knew from the dimensions of the nucleotides how far it would be from the place where the amino acid was attached down to the anticodon. And they also knew from centrifugal analysis how big the amino acyl tRNA synthetase had to be. Now remember, that's the enzyme that puts the amino acid onto the 3' end. And they discovered something very surprising. They discovered that the size of the amino acyl tRNA synthetase was such that it couldn't span from here down to here. It wasn't big enough to do that. But they knew that it had to be able to read the anticodon because it has to read the anticodon to know which amino acid to put on at this end. It's a real good example of a scientific conundrum, right? I know it's got to do it, but it's not big enough to do it. Does it have help? What does it have? Well, it turned out that when they determined the three-dimensional structure of this thing, they found it wasn't lying flat like you see here. They found, in fact, that it was bent, so that this distance in the bent direction wasn't as far apart as if this thing were lying flat. So it was really nice to see that confirmation and that conformation of the conformation, right? That what we knew from the dimensions of the molecule, there was something wrong. And the something wrong was that it wasn't a flat structure, it was a bent structure. So this is a good representation of it in two dimensions because it does show the base pairing that occurs throughout this molecule. But it's not a good representation of it in three dimensions. This is a much better one. Okay. I think I've shown you that before. I'm not going to go back through that. And the charging that goes on. So last time I mentioned to you briefly about the two different types of amino acyl tRNA synthetases. The type 1 enzymes that put the amino acid onto the 2' position, and the type 2 enzymes that put the amino acid onto the 3' position. Somebody asked me afterwards, after class, if something can be added. Does it have to be added at the very last nucleotide? Because there are 2' hydroxyls that are free throughout this structure. Is it possible it could go on to other 2' hydroxyls in the structure? And the answer is no. So it has to go on the very terminal one, and that very terminal one, you remember, is an A residue because the transfer RNA ends on a CCA. So it's on that A residue at the end where that amino acid is actually attached. As I said, the amino acyl tRNA synthetase has to be able to read the anticodon and put the appropriate amino acid on the other end. If that doesn't happen properly, then the genetic code will really have no meaning. Okay. I don't want to go into too much detail here, but I do want to show you just a little bit about what happens during that charging process. It's a little hard to see here, but this is above showing you the energy necessary to attach that amino acid to the 2' or the 3' end. That energy comes from ATP, and the product of that reaction is actually AMP. Whenever we see that happen, we know that it's a very energy-requiring reaction to put this on here, so it takes a lot of energy to put an amino acid on there. It's sort of the equivalent of 2 ATPs necessary to put that amino acid onto the end of the transfer RNA. Now, one of the things I want you to plant in your head about that transfer RNA with the amino acid on it is that that bond that links the amino acid to the transfer RNA, that bond is very unstable. In water, the half-life of that bond is on the order of about a second. Meaning that if you took a bunch of those transfer RNAs and you simply let them sitting in water, half of them would lose their amino acid in 1 second. Well, with a half-life like that, we know, of course, that the cell isn't going to let things sit around and waste energy. Cells have proteins that are participating in this process that bind to that bond as soon as the amino acid is attached. And they bind and exclude water. No surprise, because if water is present, that bond will be cleaved. Okay? These proteins, you have to imagine, are really, really important because the more of these amino acids you lose to hydrolysis, the more energy the cell is going to waste, and cells don't like to waste energy. One indication of the importance of this protein is when we look in E. coli, for example, at the protein that binds to and covers up that bond. It's called EFTU. I'll talk about it in just a little bit. EFTU, you can call it EF2 if you want, but make sure you remember it's a TU. EFTU is the most abundant protein found in E. coli. Okay? The most abundant protein found in E. coli because you don't want to have the cell waiting around for one of them to diffuse to the place where you need it. You want to have it as abundant as possible, and EFTU meets that criterion. EFTU's role, as we will see, is to not only to bind to that amino acid tRNA complex and protect it, but it's also to carry that complex to the ribosome during the process of elongation. Okay? The EF stands of the name stands for elongation factor, EFTU. Okay, so that's what's happening then during the synthesis or during the preparation for synthesis of a protein. There's one other thing I need to tell you about prokaryotic translation that's different. And by the way, I keep talking about all these things that are different in prokaryotes. The overall process is very surprisingly similar between the two. I'm going to show you the sort of steps of the process happening in translation in eukaryotes. And I'm not going to distinguish that much from what happens in prokaryotes. I'm not going to distinguish that much from what happens in eukaryotes because the overall process is in general similar. It's kind of like DNA replication. We have different names of the proteins involved between prokaryotes and eukaryotes, but the functions are very, very similar to each other. Well, one of the things that is different in prokaryotic cells is the way that translation is initiated. Okay? The way that translation is initiated. Now, you may remember I said that there's one codon called the start codon, which is AUG, which essentially starts the synthesis of all proteins. There are some minor exceptions to that, but for our purpose, we will say that that is the only way that proteins get started. Okay? AUG starts proteins in prokaryotic cells. AUG starts proteins in eukaryotic cells. The difference being that in prokaryotic cells, the very first amino acid that's put into a protein is chemically modified before anything happens. It's chemically modified. So it's not just methionine. That's what AUG codes for, is methionine. But the very first amino acid that makes it into prokaryotic cells is known as F-met. It's a 4-mil group that gets put onto methionine, and it's only the very first amino acid that has it. All the other places where methionine is coded for in the protein, an unmodified methionine is used. But the very first one has a 4-mil group attached to it. Okay? Well, why does that happen? Okay? Well, it turns out that when we study the chemistry of this process going on, it was discovered that in a prokaryotic ribosome, it's possible for the free end of methionine to form a chemical bond with the second amino acid that would actually loop it. It would sort of subvert the translation process. So prokaryotic cells evolved a way to prevent that chemical reaction from occurring, and the way that they evolved was putting this 4-mil group on there. With the 4-mil group, that looping and that bonding can't occur. Okay? So that, you say, well, why don't eukaryotes? Eukaryotes don't do that. Why don't eukaryotes do that? Eukaryotic ribosomal structure is different enough that those ends can't find each other and do that chemical reaction. So eukaryotes evolved a solution to the problem that was different, and the solution that they evolved had to do with the structure of their ribosome, not with the alteration of the structure of the first amino acid. So prokaryotes have an F-met, eukaryotes don't. How does the 4-mil group get onto there? It turns out that, remember that we have a charging reaction, right? The amino acyl tRNA synthetase catalyzes the addition of methionine to the appropriate transfer RNA. After that methionine has been placed onto that transfer RNA, okay, there's another enzyme that puts the 4-mil group onto it. So the 4-mil group is added after the tRNA has been charged. Okay? Well, cells have a way of recognizing which is the initiator tRNA, which is the one that has the F-met, and one which is internal, that is one that doesn't have the F-met. I won't go into that here, but suffice it to say that there is a way for cells to be able to distinguish those two. You can see that 4-mil group right here, or right here, as you can see on the screen. That 4-mil group is different from what was present over here. Okay. I briefly mentioned the wobble position. I want to show you something else about the wobble position that's actually kind of cool. cells, as I always talk about, are opportunists. They use every advantage that they can. Okay? And if we think about how many transfer RNAs that a cell would have, if we have 64 codons, you might expect that we would have 64 transfer RNAs, or if we exclude the 3 stop codons, we would have 61 transfer RNAs, right? Each one complementary to a different codon that's found in the genetic code. Well, it turns out that cells actually have fewer than 61 codons, okay? And the reason that they have fewer is because they actually cheat in the anticodon. This is a good example right here. Here's a transfer RNA on the bottom, and you can see the codon of the messenger RNA at the top. And you know that the anticodon forms base pairs with the codon, and when you look at this, you see, well, that G pairs with a C, that C pairs with a G, but look at that third position. The third position, which you know in the codon is variable, is base paired with an I here. I stands for inosine. Remember the inosine during the synthesis of purines? I turns out to be able to form base pairs with several things. So if you have an I at this position, this transfer RNA could form a base pair with a GCC. It could form base pair with a GCU. Okay? And I've actually summarized over here the things that I can pair with. I can pair with an A, a C, or a U. So by making a transfer RNA that has an I in that position, that's the wobble position, it doesn't have to make 3 different ones of these. It only has to make 1 with an I there. The other thing you remember I told you about base pairing in RNA is that GU base pairs are stable. So if I have a G at the anticodon end, that can pair with either a C or a U. Now, I'm not asking you to memorize all these because I don't think memorizing them matters. I think you should know that inosine, of course, gives some flexibility. But the upshot is that cells don't have to make 61 different transfer RNAs. They have to make a smaller number than that to be able to do the translation process. Okay. Let's see. What else do I want to say? Okay. Let's look at the ribosome. So this is the small subunit of the ribosome. And I like that figure. You can see in this structure the messenger RNAs shown in brown. You can see the protein parts of it shown in a sort of purple color there. And one of the first things that strikes you is that RNA occupies a pretty significant portion of that molecule. People described the function of RNA in an overall ribosome as providing a scaffolding onto which to attach proteins. That's a pretty good analogy. I'll show you a 50S in a second, and you'll see that, I think, a little better than what I'm telling you right here. Okay, we see here the 50S. Okay, we see similar sort of structure. It's bigger. Okay, and the last thing I want to show you is this guy, which is right here, which now gives a little bit more accurate picture of the overall 50S subunit. The brown is ribosomal RNA. It's remarkable. We now know at pretty good precision the structure of ribosomes thanks to X-ray crystallographers who have gotten spectra of these as a result of forming crystals and learning from that overall structure. But you can see here this overall organization and how the ribosomal RNAs provide a sort of place to attach individual proteins. The red site that's shown right there is the place where the peptide bond is formed. Okay? That's where the peptide bond is formed, right inside of there. Pretty cool. All right. Well, let's go through the steps in the process of making a protein. Actually, before I do that, let me back up. I need to say a couple things. One is that the ribosome, the small subunit, has associated with it a 16S ribosomal RNA. Okay, there are three different RNAs, ribosomal RNAs, that are found in prokaryotes: a 5S, a 16S, and a 23S. The 16S is found in the small subunit. The 5S and the 23S are found in the large subunit. Okay? You can see the size of that ribosomal RNA is about 1,650 nucleotides in size. There are 21 proteins in the ribosome, and no, I don't care that you memorize either of those numbers. Okay? The large subunit, the 50S subunit in prokaryotic cells, has both the 5S, which has 120 nucleotides, and a 23S ribosomal RNA, which has about 2,900 nucleotides. It's in the large subunit where the peptide bond is actually formed during the process of translation to join individual amino acids together. And that reaction is catalyzed by the 23S ribosomal RNA, the ribozyme that I've talked about before. The ribosomal RNA catalyzes that. So it's remarkable that all of the proteins on the face of the earth are made using catalysis by an RNA. Okay? Pretty remarkable thing. The large subunit has 31 proteins associated with it. Now, just before I came here, I discovered an error in the table, so I'll point you out the error before I show you, and that is this should say the small subunit right here. Okay? It's not everything is found in the large. This is actually in the small subunit. If we look at the individual ribosomal RNAs, we think we know the function of all of them. All right? In the case of the 5S, we think it helps to facilitate transfer RNA binding. The 5.8S is a ribosomal RNA that's found only in eukaryotic cells. So eukaryotic cells have 4 ribosomal RNAs. They have a 5S and they have a 5.8S, 2 small ones. They also contain an 18S that has the same function as the 16S. And they have a 28S that has the same function as the 23S. Now you see that the 16S and the 18S play roles in messenger RNA alignment. And messenger RNA alignment is critical for the ribosome starting the synthesis in the right place on a messenger RNA. That's very critical. If that's not placed in the right way, then the translation will not happen properly. Here's what's going on in that process of translation. There's our ribosome. Large subunit occurring on top, small subunit occurring on the bottom. There are 2 of the 3 sites I described earlier. Remember I had an A site, a P site, and an E site? There's the A site on the right. There's the P site. And the E site isn't really shown, but all that the E site does is provide an exit for the transfer RNA leaving. During this translation process, as I said, the ribosome is moving from left to right, and the transfer RNAs are coming in from right to left. Each transfer RNA carries 1 amino acid, and each transfer RNA has a unique anticodon that forms base pairs with the codon of the messenger RNA. Okay, actually, I'm not going to talk about that. That's just, you can look at that yourself. It's a general overview of the process of translation. Before I talk about starting the process, I want to say a word about the alignment of that messenger RNA in the ribosome. So the alignment is critical. One of the things that we think about starting translation is I've already told you that the AUG is, in fact, the first amino acid, the first codon that's read, and the methionine amino acid is put in as a result of that. That means then that we have to have that AUG placed in the proper place in the ribosome to start the process. Now for prokaryotic cells, that turns out to be a non-trivial thing. Because prokaryotic cells, you may recall, are polycistronic. They have multiple proteins on a single messenger RNA. Further, the first AUG, if we start at the 5' end of a messenger RNA and we start sliding in and we start reading, the first AUG that the ribosome encounters in a prokaryotic cell is not always the initiator one. So then how does the ribosome know which one to start at? Alignment is critical. And the way that alignment occurs in prokaryotic cells is through a base pairing that occurs between a sequence in the messenger RNA and a sequence in the 16S ribosomal RNA. Okay? What you see on the screen is an alignment of the AUG of a variety of genes in E. coli. And you can see that about 10 base pairs, or 10 bases ahead of that AUG, there's a common sequence. It's G-rich and it's A-rich. The consensus sequence for that is actually GGAGG. That sequence is complementary to a portion of the 16S ribosomal RNA. And it's the base pairing between those two that positions the AUG in the initiating place within the ribosome. Okay? That sequence is called a Shine-Dalgarno sequence. Shine, S-H-I-N-E. Dalgarno, D-A-L-G-A-R-N-O. It's named for the people who discovered it. Now, eukaryotic cells don't quite do that. They don't have a Shine-Dalgarno sequence. I'll show you they have some interesting sequences, but they don't align things in this way. One of the differences in eukaryotic cells is the AUG is frequently, that is the initiator AUG, is frequently the first AUG that the ribosome sees. So as long as the ribosome identifies that first AUG properly, it's usually going to work okay. Prokaryotic cells, that's not the case. So prokaryotic cells have to know that an AUG is an initiator AUG, and it knows that by the presence of this sequence here. You'll notice here there's a GUG. I wish they wouldn't put that on here, but the GUG, occasionally GUG is used as an initiator codon. For our purposes, as I said, we'll talk about AUG being the initiator. Okay, so Shine-Dalgarno is important. And I have to show you that first because when we talk about the process of initiation of translation, the Shine-Dalgarno sequence plays a critical role. Let's go through at the molecular level what's happening during translation. So imagine that we have a bacterial cell that has started making a messenger RNA. In this case, it looks like it's a finished messenger RNA, and if you want to think of it that way, that's okay too. We see this messenger RNA that has genetic code on it so that it can be translated into protein. How does that code get translated? Well, the first thing that happens in the process is that there is the binding of a small subunit. The small ribosomal subunit is bound right there. That's the sort of blobby thing on the bottom. And it binds, and the first thing that happens is that Shine-Dalgarno sequence in the messenger RNA forms that base pair with the 16S ribosomal RNA found in the small subunit. That immediately starts the alignment necessary for translation to occur properly. Another thing that happens in the process is that initiator methionine in bacterial cells, that's F-met, that initiator methionine forms a base pairing with the AUG because now it knows where to go thanks to the alignment of that Shine-Dalgarno sequence. Okay? After that has occurred, we have binding of a couple of things, and it's not really shown in detail on here, but the large subunit comes in and it sort of goes above this. It's kind of hard to draw it on there, but it goes above there to make that snowman structure I talked about before. At this point, when you have the large subunit on the top and the small subunit on the bottom, the A, the P, and the E are all set up. And that F-met is found in the P site. As I said, that's the only one where the transfer RNA starts in the P site. The very first one starts in the P site. When we have gotten to that point, okay, we then are ready to bring in the next transfer RNA with an amino acid and start the process of translation. Now, that second amino acid comes in, and it forms base pair, as you can see, with that codon that's being held in the A site for it. And the two amino acids that are carried at the end of that are brought into close proximity so that they can be joined together. And of course, the joining of them together results in formation of a peptide bond. And as I said earlier, the catalysis to form that peptide bond happens as a result of action by the 23S ribosomal RNA in the large subunit. Gesundheit. Okay? Now, we have gone through the binding, we have gone through assembly of the ribosome, we have gone through putting the first things together, the first two amino acids together. Now we're ready to move down the ribosome. We're ready for the process of elongation. Okay? We've gone through initiation, we're ready for elongation, and now we're going to spend a little time talking about how that elongation occurs. Okay, so here's where we finished the last slide. We had this guy, which was the initiator tRNA, has given up its amino acid. By the way, when the peptide bond is formed, the peptide that's there appears on the second amino acid, right? So what's happened here is we have slid down one unit. The transfer RNA that was in the P site is now in the A site. I'm sorry, in the E site. The transfer RNA that was in the A site is now in the P site, and the new transfer RNA with a new amino acid has appeared in the A site. Translocation has occurred. Now, in order to get you to this point, I need to show you a little bit about, or tell you a little bit about how that overall process occurred. So let's slow down and think about the elongation process and the translocation process, which is what has moved the ribosome one codon in the 3' direction. Okay? Well, first of all, I didn't tell you how it was that that second transfer RNA came into the A site. I just said it came and it base paired. There's actually a little bit more to it than that. So let's think about how it was, or how it is, that any transfer RNA gets into the A site. This is true for all transfer RNAs. How do they get there? Well, remember I said that one of the functions of EFTU is to carry or to transfer that transfer RNA that it's protecting to the A site. And that's what's happening. That's how the transfer RNA gets to the A site. EFTU carries all of the transfer RNAs to the A site. And it sticks the end of that transfer RNA where the anticodon is located. It sticks the end of it into position so that it can form base pairs with the messenger RNA. There's a problem. The problem is any transfer RNA it sticks in there going to be complementary to the codon? Of course not, because we've got all kinds of possibilities, right? EFTU plays a very important role. EFTU plays a very important role in determining if the right transfer RNA is there. EFTU actually determines if proper base pairing has occurred between the anticodon and the codon. So not only is it carrying that transfer RNA, not only is it protecting the amino acid from the aqueous environment so that the amino acid doesn't get cleaved off, but it's also ensuring that the proper base pairing between the codon and the anticodon occurs. If it determines that, uh-oh, I've brought the wrong one in, meaning that there's not proper base pairing that's occurring, EFTU will take it away. On the other hand, if EFTU sees that the proper pairing has occurred, EFTU will then cleave GTP, and it will leave the ribosome, and it will leave that transfer RNA sitting there in the A site. And if you're wondering how it is that the amino acid doesn't get cleaved off, the answer is the A site doesn't have any water in it. So EFTU is a really important protein. It's doing all these functions that I've just described to you. Now, one thing I didn't tell you, and I should have told you on the last slide, I'm not going to talk about detail about them, but when we look at the process of initiation, which was what's happening in that last slide, there are factors called initiation factors that help facilitate that. There are actually 3 of them. in prokaryotic cells. And I'm not going to hold you responsible for which ones are which, but you should know that there are 3 initiation factors that are necessary to start the process. And in the elongation process, there are also 3 factors that play roles in elongation. I'm going to tell you about 2 of them. EFTU is one of those. You can see the very important role of EFTU. And you notice I told you that EFTU was using GTP energy, okay, to do what it does. The second elongation factor that plays an important role in translation is known as EFG, elongation factor G. Very easy one to remember. And what EFG does is it is the thing that's responsible for moving the ribosome 1 codon away. So after the proper the proper tRNA with the proper amino acid has entered the A site, catalysis joins those 2 amino acids together. The resulting peptide is now on the incoming amino acid. The previous amino acid loses its attachment and it goes out and exits. After that attachment of the amino acids occurs, that's when translocation occurs. That's when the EFG cleaves GTP and slides the ribosome down 1 unit being 1 codon. Now, when that happens, you have an empty codon in the A site. You have a dipeptide, in this case, on the tRNA in the P site, and you have an empty tRNA on the E site. The E site is an exit site. It leaves. And now the process simply repeats itself. The dipeptide here is joined to the amino acid there, resulting in the transfer now of what will be a tripeptide onto that tRNA. Translocation occurs, this guy leaves, and it goes on and on and on. This elongation process continues until a stop codon appears in the A site. 3 different stop codons: UAA, UAG, UGA, and yes, I think you should know those sequences. When a stop codon appears in that site, there's something called a release factor, which comes in. Let's see. That's just more of the elongation process there. Okay. There's EFTU, right? And there's the protection against the hydrolysis, the structure there. And here is a release factor coming in. Release factors recognize a stop codon. Stop codons, there's no complementary tRNA for that. And when a stop codon appears in the A site, the ribosome pauses, and that's what actually allows the release factor to come in. Okay, I hear shuffling, so I'm going to end it there. I'll repeat the end part of this at the next thing. I have a fun song today, and that is the codon song. No needs amino acids, peptide bond catalysis in ribosomes, triplet bases, 3-letter codes, mixing and matching nucleotides. Who is keeping score? Here is the lowdown. If you count codons, you'll get 64. Got to line up right. Teen sRNA, shine down, gonocyte. You can make peptides every size with the proper code. Start codons positioned in the P site place. Initiate tRNAs. UGA stops and AUGs go. Who could ask for more? You know the codon, count up the codons. There are 64. All right, see you guys Monday.